Good morning, everyone. I'm Roxaray Shahidzadeh from the Office of Scientific Professional Development, Office of the Chief Scientist, and I'm pleased to welcome you to the second day of 2021 FDA Science Forum. Science as the foundation for protecting and promoting public health. Members of Cross-Agency Science Forum Working Group have volunteered countless hours over the past year to organize the Science Forum. Thus, I would like to take a minute to give a special thanks to the Science Forum Planning Committee and various work groups. All our presenters, the CDRH Studio, FDA's web team, AV team, Catherine Niab-Ursi, and Mike Kozinski, and the Cif Science Staff College for all their assistance with the virtual platform. Today, we'll have 4 concurrent sessions that presents the amazing diversity of scientific research at the FDA. Please refer to the agenda on the Science Forum website for the webcast links to your preferred session. The 2021 FDA Science Forum virtual poster sessions are also exhibited on FDA Science Forum website. The posters are available for download to all FDA Science Forum participants. In addition, the audience will have the opportunity to email their questions directly to the designated poster authors from May 26th through June 9th, 2021. Lastly, you'll receive a survey to evaluate the FDA Science Forum in the next day or two. Please take a few minutes to complete the survey as we really value your feedback to improve future FDA Science Forum. We thank you for joining us today, and now I introduce you to Dr. Carol Weiss, who will be moderating the session 6, Medical Countermeasure, Infectious Disease, and Pathogen Reduction Technology. As we also mentioned earlier, the event will offer continuing education credit for viewing the presentation. Please view the Science Forum webpage on fda.gov regarding CE for your preferred sessions. Dr. Weiss, the mic is yours.
So good morning, everyone, and again, welcome to day 2 of the FDA Science Forum. This is session 6 that focuses on medical countermeasures, infectious diseases, and pathogen reduction technologies. My name is Carol Weiss, and I work in the Office of Vaccines at CBER, Center for Biologics. I'll be the moderator for this panel. We have a great lineup of speakers. Due to time constraints, we're not going to do formal introductions, so please refer to the program for the biographies of our speakers. There will also be no live questions, but there will be one question for all the speakers to address at the panel discussion following the final speaker. So without further delay, we welcome our guest leadoff speaker, Dr. Pardis Sabeti. Dr. Sabeti, go ahead and turn on your camera and start.
It's such a pleasure to be here. I'm so proud and honored to be representing an FDA Science Forum. I'm going to be talking about pandemic response in the genomic and information age. I want to take a moment and make sure that you actually get to see my conflicts of interest, and so okay that you got more time with that. But ultimately, that's the thing I want to take the biggest beat on, is that I'm a co-founder, consultant, and shareholder in Sherlock Biosciences, and a board member and shareholder of Danaher Corporation that owns Cepheid and IDT. So these are all sort of in diagnostic technologies, and want to make sure you're very aware of all of that, those conflicts as I talk about diagnostics and part of this. And ultimately, everyone has COVID on the mind, but I want to take you a step back to perhaps a simpler time, but also a complicated time, March of 2014, when Ebola virus was detected in southern Guinea. And it came to attention in that March of 2014 when sequencing analysis done in Europe showed that this infection was Ebola, and an outbreak was declared. But it was actually traced back, right? So when epidemiologists went back, it actually is believed that the primary case, the index case, but perhaps the primary case was a small boy in a remote village in Guinea, an 18-month-old boy who had been playing in a tree inhabited by bats, who came down with the illness, but not before spreading it on to loved ones, other villagers. And by the time the outbreak was declared, it was in already in many places in Guinea. And then by April, it was in in Liberia, and then by May, when it hit Sierra Leone, it really took off like a wildfire. And if you think about this on the trajectory level, it was that, already in that early part of the outbreak, it was the biggest outbreak of Ebola we'd ever seen. But at some point, the trajectory changed. It started escalating much more quickly. And some of that was believed to be demographic, but it had to make you wonder whether or not there was anything else going on. We had a frontline view of this because there in blue is Dr. Humar Khan, the head of the hospital where we work. My team worked. We went and set up diagnostics in Sierra Leone to be able to pick this up. And we quickly identified the first case in Sierra Leone, and then sequenced 99 other genomes right after that showed that the virus is mutating. It was changing in so many different ways, so many different ways. Many of those mutations were believed to be not important, but of course now we understand that they are, and it definitely marked our attention. And the reason we share that data immediately, and one of the reasons we share that data is because it's so critical. Genomic data is so critical. It's the backbone of diagnostics, vaccines, and therapies. It can help you understand these new lineages that form and where they're moving and tie cases together. It can allow you to track and characterize the outbreak transmission, and in this case showing that it was really transmitting from human to human with one entry into the human population. And it allowed us to get to functional mutations. And so before variants of concern were things that was popular or even accepted, we were already seeing these mutations, a mutation in the Ebola glycoprotein that coincided with the change in the trajectory of the virus that appeared to be increasing the infectivity of the virus. And that is so important also because it informs public health messaging. Before, all the public health messaging was coming in from environmental exposures, but it soon became clear that this was something that required hand washing, social distancing, and other forms of contact tracing and beyond. But of course, the outbreak ended up becoming one of the largest outbreaks we've ever seen, with over 11,000 confirmed deaths. It was a tragedy, but it, where in particular tragedy because healthcare workers like Dr. Khan passed away. This was really truly a caregiver's disease. But it also could have been a cataclysm. When Ebola hit Lagos, Nigeria, a city of over 10 million, my close collaborator, Christian Happy, was there to diagnose the first case in the country. Here we are sitting at Redemption Church, which is about 100 yards away from his lab at the time at Redeemers University. This was the scene that would have happened just days. He actually had to leave. He had to leave our phone call early when he told me about what was happening because he had to get ready for First Friday that was coming in. And millions of people were descending on the campus where he was doing the testing. And we were always moved by this idea of what if a single case of Ebola moved through this crowd. And ultimately, we need better technologies, and the FDA is at the forefront of developing those better technologies to detect pathogens wherever through sequencing, to have molecular diagnostics in the hospital that can characterize a broad differential diagnosis of pathogens, and when something of great concern comes up, making sure we get those diagnostics all the way to the point of need. And we need to then connect that information in real time across the PACE, and make sure we have technologies to share that information, and not just to the hubs, the central labs, but to every individual on the planet working in coordination to get eyes on this virus and to stop it, and then to empower every actor in the system. And ultimately, through the Audacious Project, we've been working towards this effort. It's helped us to launch this effort just before COVID hit on the global scale. So those technologies to detect, those are like genome sequencing, and genome sequencing can be done in a way that is very specific to a virus like SARS-CoV-2, or it can just sequence all the metagenomics, can sequence everything in the sample to detect new viruses. But when we know a virus that we're tracking, genomics can then further inform public health. It can allow us to understand those transmission patterns, like spreading from human to human, or in the case of COVID-19, allowed us to really untangle the super spreader events to show, in fact, this is like almost a clonal expansion of the virus. To detect novel mutations like the Ebola's glycoprotein, but then early in the outbreak, the D614G mutation. And then also it can really inform contact tracing. So ultimately, we were investigating an outbreak of mumps on Harvard campus, and traditional contact tracing was good. It was able to identify certain links, but when we overlay genomic sequencing data, we could see so much more. So we needed to improve our traditional epidemiology, but overlay other technologies as well. ACE-GID, the African Center for Genomics and Infectious Disease, is a national diagnostic center where Christian Happy now works. He's taken his lab to another place. Mass Design has developed this institute, and it has this cutting-edge technology that sequenced the first SARS-CoV-2 genome in Africa and is now generating hundreds and soon-to-be thousands of genomes on a regular basis. The CRISPR technology I told you I now started, have conflicts around, but it is, I got excited about it because it's this potent technology. Essentially, the CRISPR was found in nature as a bacteria's immune system to viruses. It allows you, it allows bacteria to detect and destroy phage, viruses infecting them. And it's a simple mechanism where there's an RNA guide that, if it recognizes a particular sequence, can then go and pull down a Cas enzyme that will cause a cut. And there's a particular type of that, Cas12 and Cas13 have now all been shown to have this sort of, other features like this collateral capacity, which if it detects the thing that it's interested in, very specific, very sensitive cut, it will then essentially from there go on to cut other things in the region. If you pair that with a fluorescent probe tied to a quencher, it can signal if the original cut was made, another cut will be made to set off that fluorescence. And it's so powerful it could be moved to point of care and massive throughput. And we became really interested in this while we were studying the Zika outbreak. Essentially, during the Zika outbreak, we worked to try to help the community generate genomes. We had partnerships in 11 countries and were able to generate this data. And it was such a hard virus to sequence because, and also to detect, because it's very low copies in your blood, and it also, in other clinical samples, and it also stays around for a short period of time. But when we were able to sequence it, one thing that was impactful that we realized is in every country where we had the data to be able to date the origin of the mutation, it had been circulating for months, if not a year, before the first case was confirmed in that country. And so we needed better diagnostics. And so with this sort of new technologies, there are new technologies that are needed, are ones that are point of care that can be done and read by eye or captured by a mobile phone and shared, massive throughput that can detect lots of different things. And then we need technologies really to change our technologies to take the massive amount of genomic data that's coming in and to generate guides that can be used by everybody. And I'll touch upon the other two in a little more detail. We recently published on the bioRxiv technology that allows you to, we were able to use a lot of the massive throughput that we had to test out 20,000 guides and then use machine learning to basically figure out which guides work better, which are the most active, how do you make this better? And this is both for the CRISPR-based diagnostics, but as well as for PCR diagnostics. We're working on those technologies to also then take all of the data from diversity data that's being generated off of genome sequencing and to be able to improve that too. For the kind of quick things, essentially the CRISPR guides are highly sensitive. They can work at attomolar sensitivity. They're highly field deployable. They can work from urine and saliva, and we developed methods to use heat and chemicals to basically make it so you don't have to do extraction. It can distinguish subtypes of different variants, and we can deploy these assays, really design the guides. Actually, the longest time is really waiting for the synthesis, which we could also reduce, but having a working guide within a week. And to get those out into the field. During the Lassa fever outbreak in Nigeria, we were able to get these CRISPR technologies out in the field being used on the front lines. This kind of technology is also very well designed for high throughput. We recently published Carmen, basically pairing microfluidics and this CRISPR technology to show that on every virus for which we had at least 10 genomes, human-associated viruses, for which we could design guides, that right out of the gate in our first pass, because of that machine learning methodology we had developed, we actually were able to design guides that worked right out of the gate. And we show on clinical samples it works this well as well. And not just for detecting viruses, but for detecting subtypes of viruses as well, as we show on both synthetic as well as clinical samples for influenza. And then really now we've been working, and we're working with the FDA on a submission that we've put in for an EUA for this on the Fluidigm machine, so using the microfluidics of Fluidigm to bring this technology into the clinic right away to be able to do thousands of samples on a number of viruses simultaneously. Then we need to be able to connect that information. And ultimately for connecting, we really want to be able to connect frontline workers with actionable information. And so this goes from sample tracking and clinical data tracking, basically kind of for labs, to doing contact tracing with the community, to having dashboards and visualizations that allow you to see these things, citizen reporting that allows people to share information and get information in real time, to capture diagnostics from kind of these field deployable diagnostics, and to incorporate that genomic data. And so we need to work across the piece. I'm just going to show you a few examples of the kinds of things that we've been doing. In one aspect, we developed something called viral NGS, but a way of allowing you to do metagenomic and other kinds of sequence capture analysis in the field. And we first implemented those at our sequencing centers that we set up in Nigeria, Sierra Leone, and Senegal, and that we've been working closely with in Liberia. And then basically allowing those partners to share information in real time about what they're seeing and to be able to process that data. We've developed these global views of genomic data sets. One of the issues with most of the technologies that have been available so far is you can only really analyze a few thousand genomes, maybe 4,000 genomes, before it kind of breaks. But working with Fathom Information Design, we have this so that we have all of the genomes, over 1 million genomes, being able to be analyzed simultaneously and then explored. And so they're a terrific organization that allows that to happen. We've also been enhancing public health investigations. The New York Times recently wrote up about the work we've been doing at Colorado Mesa, and it was a terrific collaboration where we're working with a university in Colorado where most of the students, we were brought in with sort of a mission that most of the vast majority of the students were part of vulnerable communities, and the school decided that they had to bring everybody back. And of course that's not something we would recommend, but we wanted to support them and the students to do it safely. And so we were able to bring out a dashboard that we worked on with Fathom, with mobile applications that we started developing during the mumps outbreak, but essentially allowing people to share that information in real time and letting the schools see, case by case, what dorms students, infected students were in, to click on that and figure out what other groups those students participated in that were at risk, and really they didn't have the kind of money that other institutions had to test everybody every day, so they had to be smart, and that technology can really enhance that. And also to incorporate sequencing data, so we were able to then, the same dashboard has sequencing data so that you can really do case investigations in real time. And then to empower every actor in the system, ultimately, we want to be able to get those technologies out to the front lines. And so we've trained about 900 individuals mostly from West and Central Africa, but throughout the country. We've worked with the CDC. We've trained 54 individuals. This is in genomic sequencing, but through ASCID also in genomics and bioinformatics. And then this really terrific collaboration that also happened with a middle school. And I'll tell you about that a little more in a second. But this is some of the images. We had these summer-long programs at the Broad Institute in Harvard where we trained these scientists intensively every day for 7 weeks, through amazing programs. This is Operation Outbreak. Essentially, while we were tracking the mumps outbreak at Harvard, this middle school teacher reached out to me and said, I want to teach a curriculum and teach my kids about outbreaks, a civics curriculum that was going to culminate in a simulation of an outbreak. And when he sort of showed me what he was thinking, it was really delightful, just the level of commitment to excellence in this reality of the outbreak. And so as we were doing that, we were developing these models, these SIR models for small populations to study outbreaks on college campuses. And so what we did is we created an app that spreads a virtual virus via Bluetooth on cell phones to mimic and simulate a virus as an exercise for the students. And we were able to incorporate masks and vaccines and also to have diagnostics you can run to see if the sick person has the virus of interest. So we were able to incorporate all of that. And what's amazing about that is it also gives a lot of ground truth data. We were working with the FBI before COVID hit to roll this out for first responders to give them practice on how to respond by being able to mimic a real virus and see how well they detect it. And here's just, if I click again, no, so if I go back, and hit play. Yeah, so you can see, hopefully, it's not showing up for me. I don't know if it's showing up for you, but essentially, if it is not showing up, a little technical, here we go. Let's see, there. Basically, this is data from Colorado Mesa, the same school we are working with. This is individuals on the sides are ones that are socially distancing, And this was Halloween weekend. We decided to run the simulation over Halloween weekend because we thought the students would gather. Indeed, they did. What's more remarkable is they downloaded an app and agreed to be tracked via Bluetooth when they gathered in this way, on Friday night of Halloween weekend. But this is a virtual virus spreading through the population. As you might not be surprised to hear, on November 1st, a real outbreak was declared on campus. And we're using sequencing data for that. This was all done anonymously because we wanted the kids to participate openly, but it's giving us a lot of information on exactly what the dynamics were. And the dynamics are the vast majority of people are doing what they should do, but the small group of people are basically have 50 contacts, and they may be leading to these super spreader events. We also have developed a full textbook for this, and we're working with the Louisiana Department of Education. They're rolling out an entire statewide outbreak science course, and we've been working with them on the textbook as well as a partnership with Crash Course. We really want to make sure everyone is educated everywhere on how to respond to outbreaks. And finally, the last piece I want to talk about is how the idea of all of that stuff to detect and connect and empower everybody is to allow every person on the planet to help to participate to stop outbreaks from spreading. But ultimately, we need scientists to help to come up with cures, like the vaccines that you see now. Fundamentally, those vaccines are all based on the genome sequence of the virus, whether it is targeting the sequence itself or the proteins that are based on the sequence, as well as the therapies. So many of them really are ending up targeting the sequence of viruses, and so we need that information to share. And we have a number of projects that we've done to overcome. CRISPR antivirals, essentially CRISPR is designed to both detect and destroy viruses, and so we've shown that it can work in mammalian cells, and we did a series of mammalian cells, viruses in mammalian cells, influenza, LCMV, VSV, and COVID-19, really showing that you can knock these viruses down up to 300-fold. I'm going to talk more about this host cell response is the last thing I'm going to talk about. It's FDA supported and a really exciting collaboration we've been able to do with the FDA, and we're so grateful for that. But I also want to note a paper that's now in press at Cell that is on viral antigen presentation. Ultimately, what we wanted to study is, everyone knows about the proteins and what's the other kind of canonical peptides that are being presented to our immune system, but are there other things that are being presented to our immune system? Indeed, there are. Essentially, we discovered a number of non-canonical open reading frames that were presenting to the immune system and being sort of identified by T cells and creating a T cell response. In fact, the strongest T cell response to SARS-CoV-2 is a non-canonical open reading frame, something that is not thought about in our vaccines or in any way that we approach it. And indeed, a quarter of the things that are being presented to the immune system that we were looking at were from these non-canonical open reading frames. So what is all going on behind the scenes? And as genomics moves forward, we're going to see these viruses doing all sorts of things we don't understand, and we want to stay ahead of it. And ultimately, so this last project I just want to touch on is this work that we did, supported by the FDA, really trying to help as we investigate, how different therapies work and how different vaccines work. We need to understand what is the virus doing in our body. And so we recently participated in a large effort, again funded by the FDA, to do to study single cell level of COVID. But the big project that we did with you all was Ebola, really studying Ebola virus. It's a systemic, has many systemic symptoms. It's it's present throughout the body, and single cell transcriptomics allows us to see what's going on at that high resolution. So when we've been doing transcriptomics before, it's essentially like a milkshake. Everything is grouped together. All the cells in a tissue are grouped together, and it's really hard to understand like what's going on at a real level to make any meaning of it, because what we're looking at is so such a mesh. But with single cells, you can really almost like take that milkshake apart and look at every single piece of it and really understand what's going on at that level. And so this is a terrific collaboration also with the NIH's IRF, their Integrated Research Facility, where they were looking at 21 monkeys that were infected with Ebola virus. It's a real tour de force. I cannot stress enough like how challenging this was. For Lisa Hensley and I, this was a 12 years in the making of this project to get it right the way we were able to get it. And to be able to profile in vivo but also to take the cells ex vivo, which also contributed a lot for us to really understand what's happening at that cellular level. We were able to characterize cells, cell types, different cell types very, very closely to really, and to see what was happening in each of the different kinds of cells and see how they clustered specifically to cell types. But also there was clustering that we could see in the DPI, the days post-infection. The different patterns were emerging at different days post-infection, and also the amount of Ebola RNA in a particular cell. We really wanted to see like how that affected what was being turned on and off, what the different cell states were. The other kinds of things we could see is we could look at different cells and we could get a sense of what was, how many viral cells were there, viral reads were there, like how much of if you sequence out the cells and you look at all of the genomic reads in those cells, how much of it is virus? And there's a real wide variety where in some cells it was almost nothing, but in other cells it made up 50% of the viral reads were the viruses. So what was going on there? We wanted to really tease apart what was going on there, which cells were the ones that were piling up with viral reads and which ones weren't. What was driving that? And we also, we were able to see that we could investigate changes on a single cell basis, and we were able to look at the proliferation rate of different cells, and we could see that the monocytes were proliferating, that interferon-stimulated genes, go up across all cell types, and we could look at that both, we could look at that at the level of both the infected cells as well as these bystander cells that were were not infected but were connected. We could track genes that changed, with the within cell viral load. So as the infected cell viral load was going on, what was going on? And we saw that as we increased the infected viral load, STAT1 was going down, and other things were going up. Some very classic things we might expect to see, like DYNLL1 or HPS5. These are, a lot of these made sense to us, and some of them were newly discovered. So because we can now look at the very specific cells that are infected with different viruses, we could really understand at that cellular level what was being turned on in those cells that were either allowing them to be infected or, kind of like had that going on so the cells could get in more easily, or that the cells, as they got infected, the cells turned that on. And from our evidence, it looked like it was really that the virus was driving these kinds of responses. And we could, we were able to look at many of these different features. One other thing that we really did notice is, we could identify the molecular determinants of tropism, what was happening. And we really saw that these monocytes and monoblasts were a lot of infections going on there. And over time, what we saw that the viral infection was actually driving a proliferation of monocytes, but also a shift to earlier stage monocytes. That we were seeing that essentially it was shifting to these earlier stage monocytes. So all of this was being driven by the virus, and kind of these next layers is unpacking further what's going on there and understanding the mechanism of how that's happening, and then also seeing as the FDA starts to evaluate different therapies, we can now begin to say, okay, how is that therapy working, and how is it changing what's happening in these patterns and how they're happening. So it's so important to have that cellular readout to really understand how these things are working and how we might improve them. And one other piece that I didn't get to talk about, but as the last thing, is that we can also see how the virus itself is changing within the host at a high resolution. Just want to thank Christian Happy, my kind of scientific partner for 20 years now in varying capacities. All sorts of individuals. I'm an investigator at HHMI, but this is made possible. All this work is made possible by so many institutions. And that last piece I talked to you about was so fundamentally driven by the FDA. I wanted to give a particular shout out to Lisa Hensley, who was my partner in all of that work. And my lab, who makes everything possible, and that commit to excellence in everything they do, the international collaboration that motivates us and drives us and inspires us. And the patients, ultimately for all of us, it's really the patients that make this worthwhile and that trust us to do this work with them. And ultimately, thank you. Thank you for your time.
Thank you so much, Dr. Pardes. That was really exciting talk and really powerful technology. So we will now move on to the next speaker, Dr. Tony Wang. You can turn on your camera. Thank you.
Good morning. This is Tony Wang. I'm speaking from the Division of Viral Products at Center for Biologics Evaluation and Research here at FDA. It's my privilege to present some of the work we did here at FDA so that the public can see how FDA, at least some of us, are trying our best to cope with the COVID-19 pandemic. But first of all, I would like to declare that I do not have any conflict of interest in giving this presentation. Also, the content of the presentation only represents my own opinion. It does not bind or obligate FDA. So since the beginning of this year, the word variant has come into light, now becomes a term that is very familiar to the general public. But the way how CDC classifies variants, is actually demonstrated on this particular slide. And there are virtually three classifications. The first one is called a variant of interest, which we refer to a virus strain or isolate with specific genetic markers that we sort of based on prior knowledge that we believe it will be associated with change to certain traits of the virus that may enhance the ability for the virus to transmit or to escape from a prior immunity. So among those, you see quite some of these familiar variants. Those variants were circulating in the New York City or currently in India. And worth mentioning that in the UK, the B1617 now has been classified as variant of concern, so which is the second classification under CDC guideline. Variant of concern refers to a virus strain or isolate with evidence of an increase in transmissibility or more severe disease or significant reduction in escaping the immunity. So the famous ones include the B117 variant that started in the UK and the B1351 variant reported from South Africa, and those P1, P2 variants reported in Brazil. Now the last classification variant is variant of high consequence, and this group refers to variant of high consequence, which means we have a clear evidence that the existing medical countermeasures probably will either fail or have significantly reduced effectiveness against those variants. Luckily, as of today, there has been no variant of high consequences under the CDC classification. So it's quite obvious that with the rapid rollout of COVID-19 vaccine, with the public starts to feel we're really turning a corner, the pandemic appears to be going away, the emerging of these variants, particularly those variants of concerns, indeed cast a cloud over the COVID-19 vaccine efficacy. In particularly, we are, as regulators here at FDA, we are concerned how effective our vaccines or future antivirals may be effective against the more variants if there are new variants arise from some other parts of the world. So give you a little bit of piece of flavor. So here at FDA, we, as soon as the new variants were reported, we were able to run some quick assays to measure the effectiveness of convalescent plasma against the new variants. So we were able to obtain human plasma from COVID-19 patients provided by the Washington Advanced Medical Health Care here in White Oak. And then we run these assays and look at how neutralizing these human plasma are against the B117 variant or the B1351. And showing in this small figure here, indeed, although the convalescent plasma remained pretty effective in neutralizing the B117 variant, its effectiveness indeed was somewhat reduced by about 4-fold against the B1351 B.1.351 variant. So there's several important questions we're facing today. Some of them are, for instance, is the new variant indeed more pathogenic or more transmissible? And is the current vaccine protective against the variant of concern, or is it possible to re-engineer, or in other words, repurpose existing medical countermeasures against a variant of concern? So in today, for sake of time, I will present a small study to try to demonstrate how we attempt to address some of these concerns or questions. So I will be focusing on whether a new variant of concern indeed is more pathogenic in animal models. So the variant of concern we'll be talking about is the B.1.1.7 variant, which was arguably probably the most famous one in the news. So we address this question by utilizing a standardized hamster model that we have developed from the beginning of the pandemic. So using Syrian hamsters, we were able to either vaccinate the animals or directly challenge them with a variant of concern virus. Then we follow the animals for up to 2 weeks. We're able to collect different specimens such as nasal washes or blood, and occasionally we perform necropsy to look at the disease manifestation in various tissues. And we're able to observe their clinical signs, to measure their viral loads, in other words, how much virus found in the nose or in the lung or in other tissue. We're also able to characterize them, the immune response upon challenge. And lastly, we can perform histopathology analysis on these animals to indeed assess the disease severity. So the study design is such that we have 3 groups of animals, and I have to apologize. This slide actually has a mislabeling here in group 2, which I will correct here. So the group 1 animal, there are 12 hamsters, and these have been previously infected and also recovered from a very early variant, the very first one isolated in the Washington state in the US. So as I said, these animals have since recovered, they're healthy. Then we re-challenge them with a different variant. In this case, is the B.1.1.7 variant. Now, my group 2 are naive animals, which means they have never been exposed to the virus. Now, they are being challenged again here. I correct, they're being challenged with the same B.1.1.7 variant. My third group of animals, they are also naive animals, but they're being challenged with a B.1 variant, which is the one has been circulating in the U.S. since last March or June, I think up to today. And this is the parent virus, we call that a B1. So the idea is that by comparing group 2 to group 1, we will be able to tell whether the convalescent hamsters are protected against the infection of B.1.1.7, a new variant. And then by comparing group 2 and 3, we're able to tell whether the parent B1 variant versus the new variant of concern B.1.1.7 to see which one may be more pathogenic. So the results I'm going to share with you are basically shown on this slide and the following slide. And these are some molecular assays we performed. So on the y-axis in panel A, we're measuring the so-called subgenomic mRNA of the virus. These are taken, measured from nasal wash collected from the hamsters after 1 or 2 or 3 days post the challenge. So these black circles represent the specimens collected from the convalescent hamsters. The red circles represent those collected from the B.1.1.7 challenged hamsters, while the blue triangles represent those specimens collected from the New York isolate, or the B1, the parental variant challenged hamsters. As you can see that throughout the course, the viral RNA in the convalescent hamsters in the nasal wash, they're substantially low. They actually, on the day 3, they below detection limit. Well, the viral RNA collected from the naive hamsters challenged with the B.1.1.7 variant indeed was half log or 5 times higher than that collected from the New York or the B1 variant challenged hamsters. So this kind of suggests that the B117 appears to be a little replicating a little bit better, at least in the nasal cavity. However, when we measured the amount of infectious virus from the same nasal wash, this actually isn't the case. It appears that, in fact, even the parent B1 variant appears to us has a slightly higher level of infectious virus from the same nasal wash. So we don't quite understand how this has occurred yet. But then if we measure the clinical signs, particularly if we measure the body weight of these hamsters post the challenge, as you can see, the convalescent hamsters, they don't lose any weight at all. And this actually argues that they're pretty much protected probably because they don't lose weight. They appear to be healthy. While the hamsters challenged with the B117 or with the New York B1, they continuously lost weight over the course of 7 days. And at that time, we actually sacrificed the hamsters. But there's no difference between the B117 and the B1. Now, if we look at the lung pathology, so first of all, if we look at the lung consolidation score and shown in these bars, colored in red, we actually see that both the B117 and the B1, they appear to have the same level of lung consolidation. And where the convalescent hamsters re-challenged with the B117, they did not show any consolidation, actually suggesting that, again, they have very clear lung. Their respiratory function probably remain intact. Now, if we look at accumulated pathology scores, we have a system to score the pathology. The higher, actually, the worse the disease is. And you can clearly see that, again, the B117 versus B1, the accumulated pathology score remain pretty much the same. Now, to the panel E shows you the HE staining, directly show the histopathology. The convalescent hamsters, their lung looks clear. There's plenty of air sacs here. While the B117 and the B1, the lung are pretty much infiltrated with lymphocytes or macrophages. There are also a lot of overgrowth of the type 2 pneumocytes, suggesting there's severe blockage. This is the characteristics of interstitial pneumonia. And again, when we measure, we repeated the study, we pretty much saw the same thing. This is like a second study result. Again, in the nasal wash, they look at the infectivity in the nasal wash for both B117 and B1. They look pretty much the same, except on occasionally on a particular day, one could show a little bit a spike over the other one. But overall, the virus level in the lung remain pretty much the same between the two groups. The pathology score also remained pretty much the same between the two groups. So in summary, using this aged Syrian hamster models, we demonstrated that the B117 variant does not appear to be more pathogenic than the D614G or the B1 variant. I guess this is the good news, and this has now been confirmed by two epidemiology studies recently published by two UK groups using the epidemiology data collected from late 2020 until earlier this year, demonstrating that indeed the B117 does not appear to be more pathogenic. So this contrasts to somewhat reported in the news at the beginning, claiming that B117 is more pathogenic. And also, B117 does replicate to slightly higher level in the upper respiratory tract, at least for a couple of days, although its clinical significance remains to be determined. Now, the most important information we'll obtain is that the naturally acquired immunity indeed protects against the reinfection of B117. So in the future, we need to test whether B117 is more transmissible. And so this study was carried out by a number of very brief FDA scientists. Despite a very hectic schedule, we tried our best to generate useful information relevant to regulatory review as quickly as possible to inform the public. Thank you very much.
Thank you so much, Dr. Wells. Our next speaker is Dr. Xichao Liu. Please start your talk. Thank you.
Hi, good morning, everyone. This is Xichao Liu from NCTI FDA. It's my great pleasure and honor to give this presentation. My presentation title is AI Powering Precision Medicine-Based Drug Repurposing Against the COVID-19. Here's the standard disclaimer. I would like to start with the acknowledgement of the funding support by the FDA Mitochondria Initiative. The team members from the different FDA centers and offices, their expertise covers the broad spectrum of the different biomedical perspectives. Also, we get tremendous support from our sister agency, NIH and CARD, for the wet lab verification. As you know, we are running against the COVID-19. Coronavirus put the drug repurposing on the fast track. If you look at the definition of drug repurposing, it's an old drug for new use. It's mainly focused on abandoned drug candidates in the clinical trial failed other than the safety reason or the approved drug without patent issue. Compared with the de novo drug discovery pipeline, drug repurposing provides you a faster, safer, and cheaper drug development paradigm. However, the conventional drug repurposing approach is mainly based on the close clinical observation. It's mainly therapeutic in nature, so-called happy accidents. So naturally, you may expect the more systematic way to comprehensively assess the drug repositioning opportunity for the COVID-19. AI may be a solution. So talk about the COVID-19 is really a precision medicine problem. Except the major infected lung organ, some actual pulmonary manifestation has been observed among the variable population and patients with different pre-existing conditions. As you know, some of the SARS-CoV-2 carriers experience any non-specific symptoms. Some of them experience mild to moderate clinical response. Severe COVID-19 patients need a ventilator, suffer a serious clinical outcome, even death. One of the most important reasons because of the different immune response of the COVID-19 patients to the virus. So let's look at the immune response in the COVID-19. It's mainly including three stages. In the stage 1, SARS-CoV-2 engage in the innate immune system. In this stage, majority of the patients actually is no unspecific symptom, so-called asymptomatic. Once you step into the stage 2, it's really you are in the intersection stage. The adaptive immunity kicked in, generates a specific antibody, and also trigger the T-cell modulated immunity. Majority of patients in this stage will be overcome the COVID-19 without any further sequelae. And also generate the immunological memories and to further block the reinfections. However, a small proportion of the patients will step into the phase 3, where actually the patients experience very severe respiratory, inflammatory, and also suffers different organ-specific toxicity, such as the cytokine release syndrome. So once you talk about the immune system, you naturally think about another sister system, mitochondria. A mitochondrial dysfunction in COVID-19 has well reported in the different literature. However, more importantly, emphasize on the interplay between the immune and mitochondrial system may play a central role for the COVID-19 etiology and pathogenesis. Initial efforts have been made to uncover the interplay between the immune and mitochondrial system based on the domain experts and the intensive experiment. More systematic really is highly expected. So this is exactly the objective of our study, including immune mitochondrial continuum extraction, explanation, and application. So we'll first use AI algorithm to uncover the interplay between the immune and mitochondrial system at the molecular levels, and then to exploit the functional role of the immune mitochondrial continuums in the COVID-19 from the precision medicine perspective. Lastly, we will map the FDA-approved drug onto the immune mitochondrial continuums to look for the drug repurposing opportunity. So however, as you know, there's so many different AI algorithms there. What particular algorithm fulfill our needs? The deepfake actually attracts a lot of our eyeballs, may be a solution. So here's a simple introduction of the deepfake model. This model actually originally used in the imaging analysis and aims to reconstruct the face from one person to another with maximally keep the characteristics. So here's one example. Use the deepfake models and to do the face swap and to the two famous actors. One is Amy Adams, another is Nicolas Cage. The core of this particular deepfake model is they use the same encoder seamlessly and extract the hidden information from the two faces and generate high-quality reconstructed face from one to another. So this actually inspired us and to use this particular methodology to link the immune system and the mitochondrial system. So here actually is the proposal of the models by modify the deepfake models for the immune mitochondrial continuous extractions, and by using the gigantic data related to the immune mitochondrial molecular level information and also the drug transchromatome profiles. So eventually we get 21 extracted immune and mitochondrial continuums. If you look at this Tiffany graph, the 21 extracted immune mitochondrial continuums is well clustered. And also in the right figure, you will see the number of the immune and mitochondrial related genes distributed in each of the immune and mitochondrial continuums. Now the question you ask may be, what kind of the functional for this immune mitochondrial continuums, how they're correlated to the COVID-19. So in the next part of the presentation, I will use a different perspective to explain how the immune mitochondrial continuums related to the COVID-19, including such as like the functional analysis, organ-specific manifestation, disease association, severity, genetic variation, and gender and age capacity. Due to time, I will pick up several perspectives to illustrate. So first about the functional analysis. And this is fundamentally important to know what particular pathways and therapeutic targets involved in the immune and mitochondrial continuums, which provide some of the basic information for carry out drug repurposing. If you look at this circle plot graph, for each of the immune mitochondrial continuums, we list the top 5 key pathways. And due to the time, I will use this immune mitochondrial number 20 to go through the slides and to elaborate on what's going on here. For this immune mitochondrial number 20, the top 5 key pathways is listed here, such as cytokine-cytokine receptor interaction, chemokine signaling pathways. As I introduced in the immune response in the COVID-19, I don't need to emphasize more how importantly about the cytokine storm play a central role in the hyperinflammatory in the late stage of the COVID-19. So next that you know, the different patients experience the different organ-specific manifestations in the COVID-19. So it's importantly to uncover the different expressed patterns in the extracted immune and mitochondrial continuums and to provide the further useful information for the drug repurposing. With that said, we carry out the organ-specific gene set enrichment analysis. And here the bar chart shows the top 5 mostly expressed organs for each of the immune mitochondrial continuums. Go on, use the example immune mitochondrial number 20. This blue bar, actually liver is the most expressed organs. As you know, and a lot of the report has been made and to indicate the patients with specific pre-existing conditions has more susceptible to the COVID-19. Efforts have been uncovered to link the common disease and COVID-19, such as you know, like the diabetes, heart disease, all kinds of others. And here, few idols actually put on the rare disease. So we actually constructed the rare disease and the immunomitochondrial continuum network and to fill the gap. So here, actually, excuse me, there's a total of 33 of the rare disease linked to the different immunomitochondrial continuums. I continuously use that number 20 as examples. I highlight one particular disease, so-called Kawasaki disease. If you look at these cell papers, they suggest children with the COVID-19 typically experience very mild and asymptomatic symptoms. However, a small proportion of the children will experience multi-organ hyperinflammatory, which their symptoms is highly overlapped with the Kawasaki disease. One type of the explanation is the co-activation of the cytokine storm for the Kawasaki disease and the COVID-19. This is exactly our number 20 enriched pathways. So next, we'll talk about the genetic variation, as the previous presenter also mentioned this a lot. So here, actually, our efforts is put on the genetic variation of the immune-related host protein across the different population. So due to time, I just use this interleukin-6 as example to illustrate what's going on here. So if you look at these tables, I show one examples, and the genetic variation allele frequency is highly divergent across the population. Here, actually, African American has high allele frequency for this interleukin-6 specific genetic variations. That actually indicates when you develop the interleukin-6 receptors and for the COVID-19, you really need to pay attention to the targeted population. So once we get all this explanation, we are confidently to map the FDA drug to the different immune and mitochondrial continuums to find the repositioning opportunities. I just pick up one repositioning example, which actually interfere with our immune mitochondrial number 20, is dexamethasone. So as you know, dexamethasone is a synthetic glucocorticoid and mainly used to the anti-inflammatory. At this paper, Nature Review Immunology papers and point out how the dexamethasone interfere with the different immune and mitochondrial related pathways. Here I just list a few evidence. So as we point out the correlation between the COVID-19 and the Kawasaki disease, if you look at this clinical trial back to 2017, and the research has suggested use the dexamethasone to treat the Kawasaki disease, which raises another interesting point. We really need to think about how to investigate the efficacy of dexamethasone for the children with COVID-19 experienced Kawasaki-like syndrome. So next, actually, I don't need to spend more time to explain how the efficacy of dexamethasone in the severe COVID-19 patients. And if you look at the detail published in the RECOVER clinical trial and also the paper in the New England Journal of the Medicine. So lastly, I will talk about this African American again, because we point out for the interleukin-6, they have the high divergence across the population, especially for the African American. Actually, the same concern raised by the colleagues from the Johns Hopkins, they actually point out African American may have the implied response to the dexamethasone. This says if you possible use the dexamethasone to treat the severe COVID-19, but make sure in the clinical trial get sufficient enough African American peoples and in the trial, and also make sure the drug is safe and work as the way it should. So actually, a couple of the take-home message here, and I hope I can convince you the proposed deepfake models could be used to uncover the interplay between the immune and mitochondrial systems. And also, our extracted immune mitochondrial continuum is highly correlated with the COVID-19. Mapped to the FDA-approved drug on the immune mitochondrial continuums per one new way for the precision medicine-based drug repurposing. I would like to use this figure to end my slides, and AI is an effective tool for the drug repurposing. However, it's just the tip of the iceberg and does not cover all. We really need to pay attention to the different biological profiles and generate more solid results. With that said, additional verification are the must. With that said, I may just stop here. Thank you so much for your attention. I list my email here. I very welcome any suggestion, comments, and potential collaboration.
Thank you, Dr. Liu. Great talk. And next speaker is Dr. Heather Angler. Please turn on your camera.
Okay, I'm just waiting for the slides to show up. Thank you very much. My name is Heather Agler, and I'm a Senior Program Manager with our All Hazards Readiness Response and Cybersecurity team in our Center for Devices and Radiological Health. And I'm not necessarily going to be presenting research, but talking about some of the tools that you can use with medical countermeasure development and the lessons learned that I think are good for everybody to think about as we develop new medical countermeasures and protect from, future pandemics, including the development of diagnostic devices. So again, I'm in our All Hazards Readiness Response and Cybersecurity team, and our job is to respond to emergencies, but also to prepare for certain chemical, biological, or pandemic events. And so that's the focus of our group. So we have been quite busy over this past year. We triage and manage incidents and coordinate interactions both with internal and external stakeholders and evaluate and synthesize data associated with incidents, as well as coordinate the response from our center's perspective. And as I said, we've been highly involved in COVID-19, Ebola, heater-cooler, infections with heater-cooler devices, and duodenoscopes. And that's just a few of our examples. So what I primarily do within the program is support our innovation program. So the purpose is to support the development of new innovative medical countermeasure devices and new innovative devices to support the warfighter. And we work very closely with our federal partners, including DoD, BARDA, CDC, ASPR, and others, to get new medical countermeasures cleared and approved. And the idea is to be as prepared as possible for the next event. And so we want to help shorten the time it takes to develop these safe and effective medical countermeasure devices so that they are available when needed. And so during this COVID response, we've seen many different types of devices that have been critical to treating patients with COVID, diagnosing COVID. So of course our personal protective equipment was incredibly important for protecting our healthcare workers. The diagnostic tests, whether it's serological, antigen, molecular, different types of tests, different types of collecting samples, other pooled samples collection at home, and all of this was very important in the response. We also have very novel devices, such as diaphragmatic stimulators, also blood filtering devices. And of course, ventilators were incredibly important. There's a lot of attention to availability of ventilators and other types of ventilator accessories through the first half of the pandemic. So what we did with this current pandemic, it got large quickly, and we really had to think differently about how we approach the pandemic, about what we could do as a center to have the necessary products available for combating the pandemic. And so we did allow, through emergency use authorization, novel products such as these blood purification devices. We also thought differently about the personal protective equipment when we knew there were shortages out there, had decontamination systems for PPE. We also looked at novel manufacturers. So one thing that we found is that the medical device industry tend to rely on the same types of manufacturers for some of the raw components, some of the materials, and it caused problems because there was high demand across the board. And so we did work with manufacturers that are typically outside of the medical device industry, including auto manufacturers, airline manufacturers, We also reached out to the textile industry in order to make more PPE. We also saw in the news, of course, lots of the auto industry getting together to work on ventilators. And then we looked at, additive manufacturing of things like swabs and components for ventilators as well. And then, of course, there were novel policy approaches as well, including, being able to do an emergency use authorization as an umbrella that would cover multiple products. And then also issued immediately and effect guidances that would explain certain enforcement policies during the pandemic. So one of the things we really needed to do was to assess those supply chain weaknesses in order to prevent shortages and have the right devices available. And we had to analyze supply chain data from various internal and external sources in order to be able to do that. We did massive outreach to over 1,000 manufacturing sites across 12 countries, and this was just in the first part of the pandemic. We also created a group to do horizon scanning to look at what were the current treatments that were recommended for COVID patients and what type of demand that would mean for certain devices. And so with that, we were able to put in certain mitigations for things like personal protective equipment, ventilators, diagnostic supplies, infusion pumps, and others. And we did establish a Tiger team to work with our ORA colleagues within FDA to address any import issues that we saw. And we conducted outreach to component and raw material providers to predict downstream device availability concerns as well. So for shortage, when we did see that there may be a shortage or a shortage was currently going on, we did work closely with our government partners on current concerns and developing actions. We did proactive outreach to, like I said, manufacturers, also distributors. Sometimes they have a lot of data in terms of what's going on and where the concerns might be, and also group purchasing organizations that see the needs of hospitals and other healthcare centers. We also used communications as a tool through our Dear Healthcare Provider letters to explain enforcement policies or issues that we might be seeing. Our frequently asked questions were another method to get information out there, and posting of resources on the FDA webpage. So we created policies to enable greater availability of alternative products when we did see issues, and we did these through these immediately in effect guidances. And then we worked with non-traditional manufacturers to produce components, parts, materials, and even devices to advance response efforts. So this is just an infographic that we developed that looks at all the work that the center did throughout the pandemic. This is just covering 2020. So of course, all these numbers have increased, but we had a massive amount of work through outreach. We developed with the EUAs, we're receiving so many. You can see here that we received over 3,500 EUAs in 2020 and almost 2,000 pre-EUAs. So you can imagine the workload. And we, in order to make that go more smoothly, both for us and for industry, developed 11 different EUA templates that would basically explain the type of data that we would need to see for certain product areas. Certainly, with the EUAs that were issued, there were over 600. And over half of them were some type of diagnostic test, molecular antigen, antibody. And of course, we did the massive outreach to manufacturers. We had lots of our Commission Corps people deployed as well, and we responded to over 19 million emails that came. Lots of these were questions about regulatory policies, about sometimes what they could do to help if they were from other industries. And then information about the EUA process and being able to find out sort of our regulatory thinking. So of course, with our emergency use authorization, there has to be a declaration of emergency that warrants the EUA, and then it needs to be for a serious life-threatening disease or condition. We need to show basically that it may be effective and that the known and potential benefits outweigh the known and potential risks, and also that there's no adequate available approved alternatives. And we did interpret that to be either there are no other devices like that, but at times when supply chains, when we were experiencing shortages, also that there weren't enough available to meet the need. So it is a little bit different between an EUA as opposed to a medical device approval or clearance. As I said before, EUAs may be effective, and you're looking at that benefit-risk ratio, which an actual device approval will be reasonable assurance of safety and effectiveness based on valid scientific evidence. So we really do consider the totality of the scientific evidence when we're looking at that benefit-risk ratio. And we may authorize, clear, or approve a device if, among other things, available evidence shows the applicable statutory benefit-risk standard is met. I just wanted to mention we did have those 28 guidances that were in effect during COVID-19, and a lot of them explained enforcement discretion for certain changes to devices under the circumstances. A lot of those looked at ways of protecting the healthcare worker, ways of better managing patients, so allowing remote control of adjusting parameters on ventilators or infusion pumps. Also automating some of the changing of parameters such as an algorithm for oxygen titration if that device had already been studied under an IDE. So I just wanted to mention here we have the EUA, of course, that was highly utilized during this pandemic. And, you know, it's really important when you do have that EUA up there to really think about, you know, okay, my device is out there. I could be looking at collecting data on at least safety events or some sort of events that may potentially support in the future a marketing application. And so it's important to think about those as you're getting your device out there. Also, another way to have access to the device during a pandemic may be through the IDE. So setting up, of course, on a controlled clinical trial is how our IDE is typically used. Also, if you already have an IDE in process, you may be able to set up a treatment IDE, which allows you to expand access of your product to more patients. And of course, one of the tools that we definitely utilize throughout this response is expedited 510Ks. So, you know, sometimes it doesn't make sense for an EUA because you can't quite satisfy the criteria, but there is a definite need for more devices out there. Other times, it may be that the amount of data for the EUAs compared to the 510K, it makes sense just to get a fully cleared device out there. And so we have expedited many 510Ks for PPE and other types of equipment to get things out there on the market to benefit patients. So I just wanted to have one example where we utilize many of the tools that I just mentioned with ventilators and accessories. And of course, this was a big focus at the beginning of the pandemic. But we did utilize the enforcement discretion guidance for ventilators and accessories, which I mentioned earlier, which allowed remote changing of parameters, and also talked about conservation strategies for certain accessories and the use of other types of devices. We did develop communications for a Dear Healthcare Provider letter that said what to do when supplies are low and provide conservation strategies for accessories, a website that explained our current policies and our frequently asked questions, which were based upon what we were receiving within the center, that we wanted to get the information out there for others to hear as well. And then we did have our EUA for ventilators and accessories. This is one of our umbrella EUAs. And as of April, I believe we had 118 ventilator and accessories authorized under that EUA. We also worked with a unique organization, VentConnect.org. We worked with this organization, which was basically comprised of the aerospace industry, to match them up with ventilator manufacturers when they were in need of certain components, so that they could source them outside of their typical supply chains and do it in a way that also complied with FDA regulations. And I just want to mention our diagnostic test. That's been an incredibly important tool, and it's been developing, of course, in terms of the types of applications for these devices. You have your mainly molecular antibody and antigen tests. We also looked at collection of samples too through home collection, pooling of test data, point-of-care devices, multi-analyte, and of course at-home testing as well. The lessons learned for the medical countermeasure development during COVID-19. First, I think it's good for manufacturers and developers to think outside the box regarding materials used. A good example is gloves, where everybody wants nitrile gloves. And so all of the gloves in the market are dependent upon nitrile. And so, if we can think about different materials and qualify different materials before these emergencies happen, it would be incredibly beneficial. We also have that same issue with gowns and N95s. They're made out of the same material, and so there was a real issue with availability of that raw material. And to think creatively about manufacturing and medical countermeasures. During an emergency, these supplies need to ramp up quickly, and we definitely experienced this. And, how can we utilize tools like additive manufacturing to make products quickly? And what about on-demand manufacturing, developing capabilities where maybe on-site you could be producing N95? And then to think proactively on how to collect data from devices that are being used during the COVID-19 to support full approval or clearance. So think about these ahead of time. When there is an emergency, think about how you might be able to use real-world data if your device is on the market. And small components and raw materials definitely play a critical role in the ability to respond, so we emphasize supply chain resiliency. So I just wanted to thank you all for the time and to be able to talk with you here today. My email address is there under general questions, and I would certainly welcome you to get in touch with me if you have any questions. Thank you.
Thank you so much, Dr. Engler. And our next speaker is Dr. Catherine King. Please turn on your camera.
Good morning. I'm Catherine King from the Office of Biotechnology Products, or OBP. And a quality product of any kind consistently meets the expectations of the user. Drugs are no different. Patients expect safe and effective medicine with every dose that they take, and quality is assuring that every dose is safe, effective, and free of contamination and defects. It's what gives patients confidence in their next dose of medicine. Today I'll be talking about the freedom from adventitious agents contamination. But before I get going, I'll say that the views reflected in this talk are my own and should not be used in lieu of regulations, published FDA guidance, or direct discussion with the agency. So products regulated by OBP include recombinant therapeutic protein products like monoclonal antibodies. And these must be shown to be free of detectable adventitious agents. Adventitious agents are microorganisms like bacteria, fungi, mycoplasma, and viruses, exogenously introduced during cell line development or manufacture of a biotech product. I've highlighted viruses in light blue because that will be my main focus today. Why are we concerned? Well, patient safety is intricately linked to drug product quality, and we don't want to expose our patients to pathogens. So the majority of OBP-regulated products are manufactured using cell substrates as a means to express recombinant proteins. While cell substrates may be the source of a contaminant, such as a virus, they also provide a great environment for the growth of adventitious agents which have been introduced from raw materials, personnel, or environmental sources. So patient exposure to contaminated biological products is not new and actually goes back to mid-last century. Example of this is the finding of SV40 contamination of primary rhesus monkey kidney cells used for the manufacture of live and inactive polio vaccines. Subsequent to this, it was mandated that vaccines be free of extraneous microbial agents. In the ensuing years, a testing panel has been built up, which is performed on all products, including the products regulated by OBP, and has served us well as an industry in terms of maintaining a safety record. However, in 2010, porcine circovirus 1 was discovered by an academic lab in a licensed product. What's interesting about this is that it went through the traditional testing but was not detected. It was detected by using new molecular methods, which I'll come to later in this talk, indicating that there are gaps in our current testing methods. However, even if you do detect an event, this is still a problem. So a virus contamination may manifest itself in a massive cell culture collapse, so at least you know it's there. Or it could persist with subtle signs spreading through your entire downstream process. Either way, you need to stop production, carry out a root cause investigation, decontaminate, and fix your problem. All of this takes time. And depending on what product supplies you have on hand, this could lead and has led to drug shortages, which are also a public health concern. So overall, virus contamination in biotech manufacturing is a rare event. There's a nice reference which has been published here, which was from a closed consortium representing 3/4 of the global mammalian cell culture capacity. And in a survey conducted by them, as well as publications from the literature, a total of 26 contamination events were noted over 36 years of manufacturing experience. If you think of the number of bioreactor runs in this time, this is a relatively good safety record. However, the finding of PCV1 in a licensed product by broad molecular methods indicated that there are gaps in our current testing methods. Another reason that we might be interested in new methods, in addition to filling the gaps, is a faster time to results. So in 2012, 21 CFR 61012 was updated. It was to remove the specifics of sterility test methodology from the CFR. And the idea behind this was to encourage biological products manufacturers to use state-of-the-art methods. An example of this is the traditional sterility test takes 14 days. However, rapid methods like membrane filtration plus ATP bioluminescence can reduce this time to 5 to 6 days, which is a substantial improvement. Likewise, traditional mycoplasma testing is a lengthy procedure. It can take up to 28 days to detect these common contaminants of cell culture, some of which are human pathogens. In our office, we've seen and accepted commercially available test kits as well as in-house assays. These assays target 16S rRNA and you may do your amplification directly from your in-process intermediate, or you may include a biological amplification step in order to enhance sensitivity. Either way, results in hours to days is much faster than the 28 days of the traditional test. We have accepted these if they've been appropriately validated and shown to be sufficiently sensitive. To that end, they should be comparable or better than the traditional assay. They should include relevant mycoplasma strains, including non-cultivable, with a justification of your choice. In addition, you should include positive and negative controls as well as an extraction control. Like mycoplasma testing, traditional virus testing is lengthy in nature. In particular, the broad assays to detect the unexpected viruses take weeks to result. In addition, the assay must be susceptible to infection by the virus. The virus must be able to replicate in the assay, and an observable effect must be produced in order for a positive result to be seen. Therefore, in addition to the issue of time, there are gaps in the testing. So a conversation surrounding the broad molecular methods for virus testing stemmed from a 2009 cell substrate workshop. And one initiative that came out of this workshop is Technical Report 71, which focused on 4 broad methods for virus detection, which are listed here on the screen. In 3 out of the 4 methods, the user is boxed in by the bioinformatics associated with the primer or probe set used in the assay. And for this reason, as past contamination events have shown, while the same culprits do appear time and again, it may not always be the case. And this is why it's important for us to include broad, unbiased assays in our virus testing panels. And so for this reason, next generation or massively parallel sequencing has really come to the fore as the prime method of interest for non-targeted virus detection. And to that end, in 2012, an advanced virus detection users group was established at the Parental Drug Association, which transitioned into an active users group in 2014. This group serves as a forum for scientific discussion on the organization of collaborative studies and the generation of resources for the public. In addition to this group, there have been multiple public meetings held with PDA or other organizations to discuss the idea of using NGS for virus testing in biologics. So molecular methods for virus testing for biotech products have been in use for many years. A prime example of this is the specific PCR test for the minute virus of mouse. So specific PCRs clearly lends itself to lists of known viruses. One example of this is the rodent antibody production test listed in Table 3 of ICHQ5A. In the traditional state, these are in vivo assays in which the animal model is inoculated and following an appropriate incubation time, checked by immunological endpoints for specific viruses. Obviously, it would be possible to use specific PCRs for this list of viruses. However, another approach that we've seen is the use of multiplex PCRs, in which primers are designed to conserved regions of virus families And then sensitivity may be amplified by using nested primer sets, and any amplicons would be detected on the basis of size. And in our office, we've accepted similar methods for IND submissions. So next generation sequencing is any technique whereby a huge volume of sequencing data is collected and analyzed. It's collected without sequence bias. There are a variety of platforms available, and the mechanics of each of the platforms differ. However, broadly speaking, you start with nucleic acid extraction. Once you have your nucleic acid, you RT your any RNA to make cDNA. Depending on what your sample is and your intended endpoint, you may wish to include steps to reduce sample complexity in order to increase sensitivity. You may wish to fragment your sample in order for your library preparation. Once you have a library prepared, you sequence it, and the output of the sequencing is run through your bioinformatics pipeline against a curated virus database. It's important to have a strategy for following up on any positive hits. So as this method involves many steps and is technically challenging, ultimately it will be important to show that each of the steps has been appropriately validated for its intended use. And the importance of well-characterized resources and standards for NGS is highlighted by the following study. There are a lot of details on this slide, and you can read the reference yourself. However, in short, there were 15 labs that generated data for this study, and they were looking to see if they could detect viruses in a highly complex virus stock, which consisted of 25 target viruses, as well as some additional non-target viruses, which were found by some of the labs. 3 of the labs were able to detect all 25 viruses. The majority of the labs detected 20, while certain labs only detected 6 viruses. Certain viruses were detected more consistently than others. What this study shows is that while NGS can be very sensitive, the application is not always equivalent in its ability to detect viruses or certain types of viruses. Therefore, it's important for reagents and tools to be available for folks performing NGS, and also for people, the end user, to consider the many factors which can impact upon its ability to detect virus. So attaining a good quality starting nucleic acid sample is critical for overall assay performance. As we're all aware, different viruses have different physical chemical characteristics, which may impact upon the ability to obtain good quality nucleic acid. Therefore, it's important to validate your extraction method you're using, impact of the sample matrix that you're in, any treatments to reduce sample complexity. Should you have a low amount of material, the impact of any global amplification steps, that you have sufficient quantity and quality of starting material, and the right size fragments if this is relevant to your platform. Virus spiking studies can help understand if there's some bias against specific type of virus in the first place, and regular inclusions of extraction and treatment controls can help assure the assay is behaving on a routine basis. In addition, you need to consider your platform, the read length hereafter, and the number of reads or depth of sequencing. Other considerations include bioinformatics, the pipeline you're using, the database you're using, and follow-up strategy for positive hits. As I mentioned, as a users group, which serves as a forum for discussion of scientific issues, I'd like to draw your attention to some literature references which have come out of collaborative studies from this group. These 2 references listed on the slide were driven by a ResearchCons lab here at CBER. In the first of the 2, common virus stocks were used with a common cell substrate for generation of the different matrices. Other spiking studies have been ongoing, and we're eagerly awaiting publications of the results. And in addition, the last reference is an expanded 8-lab study, which was used in support of proposed international reference reagents, which have been adopted by the WHO, and these are shown here. So these 5 viruses shown on the slide are the WHO CBER International Reference Reagents. They're available upon request from ARISACON at CBER for qualification and validation of NGS, or with justification for other purposes that may advance our understanding of NGS capabilities, such as comparison to conventional assays. So other resources that have been developed include the reference virus database, also generated by the ConLab. The reference is up here for it. It's a comprehensive virus database in which cellular content has been reduced. It's nucleotide-based and updated regularly and hosted externally at the University of Delaware, so it's publicly available. And this database was used as the template for the generation of a protein version of the reference virus database by the Elwha lab at Institut Pasteur. This is also updated regularly and hosted externally, so it may be used by anybody. So regarding application to OBP products, with respect to NGS, we've had emerging technology team interactions concerning non-targeted approaches aimed at replacement of the in vitro indicator cell line assays. In respect to these non-targeted approaches, we've also seen people wanting to include stepwise steps to implementation, which include the introduction of a targeted phase with trope-relevant viruses. And with that, I'd like to thank the following individuals as well as the organizers for allowing me to speak, and thank you for your time.
Thank you, Dr. King. And our next speaker is Dr. Elizabeth Miller. Please turn on your camera.
Hi, can everybody hear me? All right, good morning, and thank you to the organizers again for putting on this incredible science forum and specifically for hosting this session today. I'm Elizabeth Miller. I'm the Assistant Commissioner for Medical Products and Tobacco Operations. I returned to the agency 14 months ago, and I lead the inspections program of our biologics products, bioresearch monitoring, medical devices and radiologic health, pharmaceuticals, and tobacco operations. I'm honored to be with you today along with so many of my esteemed colleagues, and particularly to represent FDA's Office of Regulatory Affairs, or ORA. To say it's been an extraordinary time to be at FDA is a grave understatement. Despite many challenges and constraints, it's been fantastic to be back at the agency to see the heroic efforts and innovative solutions that our mission-driven workforce has brought to this public health emergency. This was a time like no other for both FDA as well as regulated industry. And today I'm pleased to share some information on the novel regulatory framework during the pandemic, including our investigation efforts to evaluate manufacturing operations for vaccines and therapeutics to continue our commitment for upholding our mandate to protect and promote public health and ensure the products we regulate are safe, effective, and quality for patients and consumers. This morning I'm going to provide an overview of FDA's field investigator activities as they support the agency efforts with medical countermeasures and emerging technologies. I will address briefly the regulatory framework that we are working under as we evaluate manufacturing and related operations, including packaging facilities. I also have some case studies to share, and we will touch on some lessons learned from our investigators. And lastly, I want to share some of our future work and the roles we see expanding as we have had opportunities to reassess, fine-tune our efforts with medical countermeasures. Under Section 564 of the Federal Food, Drug, and Cosmetic Act, once the Secretary declares an emergency, the FDA Commissioner may authorize the use of unapproved medical products or unapproved uses of approved medical products in an actual or potential public health emergency. These medical products, also referred to as medical countermeasures, are drugs, devices, and biologic products intended to diagnose, treat, or prevent serious or life-threatening diseases or conditions caused by chemical, biological, radiological, or nuclear agents, including emerging infectious disease threats where there are no adequate approved alternatives available. The Office of Regulatory Affairs is the FDA Medical Countermeasure Initiative partner responsible for protecting consumers and enhancing public health by maximizing compliance with FDA laws and regulations. ORA inspects and investigates regulated products and facilities and conducts compliance and enforcement activities. In March 2020, like the majority of the world, we had to adapt our operations. We made the decision to postpone routine foreign and domestic operations. Notably, all work deemed mission critical was assigned and was accomplished. While the majority of our mission-critical inspections have been domestics, we have successfully conducted foreign mission-critical inspections in nearly 30 countries. And this has not been without tremendous effort to mitigate risk to our investigators and has entailed tremendous fortitude on their part. Many of them have had to quarantine upon arrival in foreign countries, including 14-day confinements in military housing or hotel rooms, where in some cases they were not freely available to move about, and they had their food delivered to their room. In last July of 2020, we resumed our prioritized domestic surveillance inspections. We were able to do this using data. FDA developed an advisory tool as one factor to help us determine when and where it's safest to conduct prioritized domestic inspections. The COVID-19 advisory rating system uses real-time data to qualitatively assess the number of COVID-19 cases in a local area based upon state and national data, using metrics including phase of the state and statistics measured at the county level to gauge the current trend and intensity of infection. I want to note that inspections, the physical on-site evaluations, will always be an essential part of how we evaluate operations to verify that manufacturers maintain current good manufacturing practices in their day-to-day operation. But during this time, we've also learned the inherent value in employing a suite of multiple tools available to us, and the need to develop new tools that can provide additional regulatory information when constraints exist due to disasters, whether these are natural or of an infectious nature, and the resulting damage to infrastructure and restrictions on the ability to travel. So my programs are organizations that provide the boots on the ground. Our field investigators not only conduct inspections of existing products and facilities, but we also get called upon to evaluate new products or facilities. When it comes to assessing new technologies and new facilities, we have available an opportunity for regulated industry that is a program entitled the Pre-Operation Review and Visit Program. This program is described in FDA Field Management Directive 135, whereby the agency can be requested to provide comments about the construction plans for new facilities or major modification of an existing facility during certain construction phases. The program was developed to provide specific outcomes and may be advantageous to both industry as well as us as regulators for a number of reasons. Not only does it have the potential to reveal possible defects early, preventing costly construction errors which could lead to unreliable operations and products, it also provides the agency with the opportunity to become aware of novel and evolving implementation and use of technologies and platforms. Early involvement will increase efficiency and the results in timely processing of applications. The purpose for the field review is to offer the best opinion on whether new or modified facility and processing procedures would comply with current good manufacturing practice regulations. Pre-operational review should not be confused with Center for Drugs and Center for Biologics site visits. Site visits are typically designed to offer experiential and first-hand learning opportunities to FDA staff involved in the development of FDA quality metrics program, and to provide stakeholders with an opportunity to explain the advantages and challenges associated with implementing and managing a robust quality metrics program. One goal of these site visits is to provide FDA staff exposure to existing quality metrics programs through a tour of operations and discussion with establishments to assist staff in further developing the quality metrics program. Another goal is to provide a forum for industry to engage in the process and provide additional feedback to improving our quality metrics programs. So let's discuss the emergency use authorization process. With the declaration of a public health emergency under Section 564 of the Food, Drug, and Cosmetic Act, FDA may authorize unapproved medical products or approved medical products to be used in an emergency to diagnose, treat, or prevent serious or life-threatening disease when certain criteria are met, including no adequate approved or available alternatives. Section 564A, as added by the Pandemic and All-Hazard Preparedness Reauthorization Act, established streamlined mechanisms to facilitate preparedness and response activities involving certain FDA-approved medical countermeasures without issuing EUAs, which is resource-intensive as a process, and these authorities apply only to FDA-approved eligible medical products intended for use during chemical, biological, radiological, nuclear, and explosive events, emergencies. And these provisions include, the ability to empower FDA to extend the expiration date of eligible FDA-approved medical countermeasures stockpiled, permit FDA to waive otherwise applicable current manufacturing practice requirements for storage or handling or to accommodate emergency response needs. They allow emergency dispensing of MCMs without requiring individual prescriptions or all of the other information that's usually required for labeling for each recipient of the MCM, and also permit the Centers for Disease Control and Prevention to create and issue emergency use instructions concerning the use of FDA-approved conditions of use for eligible products. Notably, during this pandemic, many EUAs are underway, as well as many have been authorized. Recognizing that this pandemic would require a thoughtful and deliberate approach to our oversight of firms producing medical countermeasures where none were previously available, in my office, the Office of Biological Product Operations, in partnership with the Office of Pharmaceutical Quality Operations, launched an EUA investigations program in the fall of 2020. This included the development of a field management directive directing the conduct of EUA investigations. As a reminder, EUA allows unapproved medical products and approved products with unapproved uses to be introduced into interstate commerce if they may be effective during a declared public health and national emergencies when there are no available approved alternatives. So this new Field Management Directive, FMD 153, outlines the process that investigations of facilities manufacturing potential EUA products could be assessed. Keep in mind our authority to issue EUA is separate and distinct from the use of medical products under an investigational application. For example, an IND or an IDE, which is an investigational new drug application or an investigational device exemption. What the field organization outlined in this Field Management Directive is the information gathering activity to ensure that potential facilities intended to manufacture EUA products are capable of manufacturing, packaging, labeling, or testing the product intended for use in a public health emergency. The purpose of the investigation is to ensure the public health goal of providing access to safe, effective, and quality novel critical therapies or diagnostics. I just want to move on to highlight a couple of the statutory context of drugs inspections. The FD&C Act in Section 510H reads that we shall inspect drug manufacturing facilities registered with the agency. This would also apply to the extension of manufacturing facilities such as contract labs, contract sterilizers, as well as packaging facilities. Section 351 of the Public Health Service Act and Section 702 and 704 of the Food, Drug, and Cosmetic Act provide the regulatory authority for FDA to conduct investigations and collect records at establishments for medical products, including those that may be subject to an EUA are manufactured, packaged, labeled, or tested. And practical aspects of an inspection under 704A1 read that we can review anything bearing on whether a drug is adulterated or misbranded, except financial information at establishments in which a product is manufactured, processed, packaged, or held. 704A4 reads that we can request records and other information in lieu of or in advance of an inspection. This applies to drugs and biologic drugs, and this authority does not extend to remote interactions. And in our drugs and biologics programs, we have embraced and used this authority under Section 704A4 to request records in advance or in lieu of inspections. Although we've had this authority since 2012, during the pandemic, we have implemented it broadly. It applies to human drugs and biologics, and it applies in the context of pre-approval inspections, surveillance inspections, as well as following up to defect reports. So now some real-world examples of some of the work we've done supporting emergency use authorization. Typically, the process starts when the centers, either CBER or CDER or other, CDRH, are identifying to ORA that the facilities identified in a submitted EU proposal need potentially to be evaluated. In a number of these recent investigations, we supported the review of an EUA by doing actually on-site investigations. This is designed to assure quality of the products and ensure that the quality systems are in place and to show that the facilities producing the EUA have those quality systems. The first one I want to discuss involved a new product being introduced into an existing multi-product facility. The evaluation involved the understanding of the building and facilities, and it started with the HVAC system and controls. Investigating and understanding the location of the HEPA filters and focusing on the assessment of air exchanges, room pressurization, and cascade of air and air changes in the manufacturing process areas was included. One of the key areas for the new product introduced into the multi-product facility is to evaluate the potential for cross-contamination. And this gets to assessing what other products are in the facilities, and how the facility ensures cleaning and disinfected as they are changing over. We have expectation that risk assessments be performed for the analysis of the impact to a process and complexity of the systems, and a thorough hazard analysis of critical processes is conducted for all stages. Material handling is another essential area for assessment during these investigations, and samples are analyzed to ensure multiple quality aspects, including the identity as well as other attributes, including impurities, endotoxins, and other quality limits are meeting and comporting to predetermined specifications. The outcome in all EUIUS investigations has been informative not only to FDA but also to industry, as it provides reassurance that the controls at the facility at the time would facilitate good quality control for essential medicines being produced. So going on, I wanted to note that we have had other interactions. One came where we were asked to do an investigation where we were looking at storage and handling conditions with a vaccine that was being produced abroad. The authority abroad had some constraints with their regulations, and ORA stepped in to conduct collaboration among
And I think we lost the audio, and Dr. Miller. Dr. Miller will come back in. Thank you for your patience.
I'm sorry, can you hear me? My phone cut out. The battery died. I will just say with that, as we continue through the public health emergency, our goal for EUM investigations is to ensure that our facilities that are identified in these applications for essential products have appropriate quality systems in place that are needed to produce these essential products, and that we will continue to use our oversight capabilities to ensure that their quality is safe and effective. And I thank you for your attention and apologize for the disruption in my phone.
Okay, thank you very much. No problem with the phone battery dying. That's happened to all of us. So again, I want to thank all the speakers for really a set of terrific talks. They were covering a range of methodologies and technologies that really all intersect in efforts to create safe and effective medical countermeasures for pathogens. So we're going to start now with our panel discussion. All speakers should have their cameras on, and they've all been given a question to think about, and we'll have 2 minutes to answer. It'll be answered in alphabetical order, and I'll call each speaker in turn. And so the question is, in your area of expertise, what are the biggest scientific or regulatory challenges for the development of safe and effective medical countermeasures against pathogens, and ensuring that they are free of adventitious agents? As well, what innovative technologies and methodologies have great potential for addressing those challenges? So we will start off with Dr. Engler, please.
Thank you. I think I talked about some of these lessons learned and concerns, and I think one of the biggest challenges is understanding the supply chain and the raw materials. I mean, what we found is that even if people had multiple suppliers of particular material, all medical devices tended to rely on those same suppliers. And what we really need to stress is that people need to think about having a resilient supply chain, about diversifying, about thinking about, if my device is meant to, be used in a pandemic or an emergency event, so typically it's going to have this very steep increase in demand. And to think about that ahead of time, think about different manufacturing techniques, think about ways of diversifying to make that supply chain resilient. and think of innovative ways of how you can manufacture, on-site or on-demand manufacturing are some of the ideas that I think would be incredibly helpful.
Great. Thank you. Next, Dr. James.
Okay, so I'll just reiterate, I believe the industry has had a very good track record, but I think the biggest challenge in broad virus testing is that you don't know what you're looking for. Hopefully nothing is there, but it's important to know that you'd be able to capture it if it were there, so looking for the unexpected. In terms of innovative tools, I believe that non-targeted NGS has a great potential for filling in the gaps left by the traditional assays and could be used as a supplemental assay. While strides have been made, we're still at the point of generating data needed to support it for use as a replacement for the routine indicator cell line assays, which seems to be the major goal of interest, at least amongst the ETT packages that we've been receiving as an office. There are many ongoing group spiking studies as well as individual studies, and I think the output of these studies will be informative. It's also important to remember if you're considering replacing routine in vitro tests, it will be important to show that the assay you're using works as well or better than the current assays for you. Thank you. Great.
Hi, this is Charles. So I think it's very important and about the translational power about the emerging technologies, and also make sense of the results and for the regulatory applications. As you know, if we look at the trajectory of any emerging technologies, we first improved tremendously, people feel very excited, and then we get some of the controversial results and people depress a little bit about technology. Eventually, we need to lay out the pros and the cons of the new emerging technologies and position that into the right application domain. So this is exactly to adopt any of the emerging technologies. This is why from the artificial intelligence perspective, our center actually newly established the Air AI Research Force and try to fill the gap and working closely with the reviewers and look at the different biological profiles and facilitate this process and to make the AI really adopt and to the regulatory application and real-world applications. Thank you.
Thank you. Dr. Miller, please. Dr. Miller, can you...
Your microphone. Hi, thank you. The greatest challenge for ORA was a regulatory challenge and really involved creating a good balance between ensuring public access to products that were newly developed to meet unmet dire need, with the assurance that such products were produced in a means that helped ensure they were manufactured to achieve quality. As discussed, the number of unprecedented EUAs being received by the agency really needed to direct us to develop how we would conduct our work to achieve this balance. And the new FMD really provided our investigators with instructions when conducting an investigation by a facility engaged in the manufacturing, packaging, labeling, or testing of these EUA products to, appropriately facilitate this access urgently needed in a declared public health emergency. These activities allowed us to directly observe observations of the production of the COVID-19 vaccines, as well as other medical countermeasures, to ensure that firms had proper controls in place and ensure the safety, purity, and potency of these. We provided feedback to firms to optimize their compliance with CGMPs and to ensure that patients and their healthcare providers would have access to safe and effective medical countermeasures. Thank you.
Thank you very much. And Dr. Sabeti.
Well, first I want to just take a moment and say, all the things FDA did right this year. I mean, what an incredibly challenging year to, have to deal with a unfolding pandemic and to deal with everybody and their mother developing a diagnostic test and hundreds of vaccines coming through. And so, I mean, just a remarkable feat. I think it really shows a lot of promise what is possible. You know, and it's gonna remain a challenge that as these viruses emerge, they'll emerge quickly, and, you know, out of nowhere. And you're gonna a big thing is how to get the clinical resources to bear for folks to quickly start doing that test, and to help a lot of individuals who may be new to the FDA to kind of go through that regulatory process. As someone who's submitted their first FDA thing, the document is difficult, and you have to take a moment and try to navigate it. So I would call on the U.S. government to give more support, to have more. I know how hard you all are working. The regulators are working to review this, so there needs to be a way more support to get more people to review this, to also help handhold us technologists in getting it through, to get those clinical specimens where it's possible if a new virus emerges. If it's not on your coast yet, you want to stop it from getting there. So how do we get those samples out to those people so they can start testing their technologies as well? So there's a lot of work that needs to happen to allow us to be as nimble as possible. That comes with more resources to the FDA to both do the regulatory oversight, but then also to help the great partners around the world who are just new to the system, if there's a way of doing that. So yeah, I think a lot of that is just a lot of really wonderful work that's been done so far, but way more investment in allowing folks, lots of new partners to come to bear and to work. And the one other thing I'll mention is also from the standpoint of the NGS and these other technologies that are trying to be massively multiplex, like the one we put forward, our biggest challenge is actually we could only try to get an approval for things that we had clinical samples, or we did contrive and there's other work, but having better clinical sample resources so that we can also develop diagnostics for things that are not here yet, or or like a broader set of things is another thing that will be incredibly critical. So there's a lot more to support, but there's also just a lot to embrace and to congratulate.
Great, thank you. And Dr. Wong.
Yeah, so to me, the biggest hurdle, in fact, as a vaccine regulator is we still don't have an established immune correlate that predicts vaccine efficacy. So we're still relying on the old-fashioned way of evaluating a vaccine efficacy through clinical trials by looking at the clinical outcome after vaccination. So this process is long, laborious, and difficult to evaluate. You need a lot of expertise to do that. But to give the credit that last year really took just about 8 months for the vaccine manufacturers as well as FDA to go through all these hurdles, and finally we got vaccines issued with EUA. I mean, that is already record speed. But for long term, I think, especially in the future, if we want to cope with a emerging pathogen like SARS-CoV-2, it would be nice if we have a way to evaluate vaccine efficacy quicker. And as to the variants, I think we need a technology that can recapitulate human disease much better than the ones we have now. So that requires a lot of R&D in developing those tools, everything in combination. Thank you.
Okay, terrific. Once again, I really want to thank the speakers. All your areas of expertise are so critical for developing safe and effective countermeasures, and in pandemics and emergency settings, really important to think with innovative approaches, and I think you've all demonstrated that. So we're going to close this session now. We will have a break for lunch until 12 o'clock. There will be 2 concurrent sessions in the afternoon. Please refer to the Science Forum's website for the webcast links to the sessions that you prefer. And be sure to visit the 2021 FDA Science Forum virtual poster session. So once again, thank you all, and have a good lunch.